So this is like, so how does the cell communicate with each other? So basically, ligand will bind into this receptor, and then this receptor undergoing a conformation change. So instead of like this shape, it changed to this straight shape. That's called the let's call the ligand and the receptor working together, and then it's the receptor will from inactive into the active activated forms. So so cells contain different cells surface receptor that respond to extracellular signals. So we're gonna talk about each one by one. So most of the signal molecule are either like small hydrophilic molecule or large molecule that don't readily pass through the plasma membrane of the cells. So basically, so this is signal molecule. Most times they are like hydrophilic molecule. Hydrophilic, which means it's more like a polar molecule. It's like water. So most of the times, this kind of receptor, like proteins like are composed of amino acids. So most of the times, the protein surface, they are hydrophilics. So hydrophilics molecule will have good interaction with hydrophilic ones. So that's why those signal molecule, most times they are like small hydrophilic molecule, or they would also could be like a large molecule. So those large molecule is like a protein, like protein and protein interaction. That's like happen a lot in a living organism. So basically this molecule, signal molecule called extracellular signal molecule, they will bind to cell surface receptors found in the plasma membrane. So a typical cell would have like a dozen, even 100 different cell surface receptors, and enable a cell to respond to the different kind of extracellular signaling molecules. So basically, this type of a receptor can be divided into three types: enzyme-linked receptors, G protein-coupled receptors, and ligand-gated ion channels. So, like for example, so this is a good example about enzyme-linked receptors. So a signal molecule will bind to this kind of enzyme-linked receptors. So and then when it means enzyme-linked receptor, so basically, when a signaling molecule they bind to these kind of receptors, like I said, its conformation gonna change, and then because this conformation gonna change, this receptor will convert from inactive form into the active form, and then this then like before it's like this, now it's like this, become the change information, change the conformation, and then the receptors, because it changed the conformation, so it can become the catalyze some kind of reaction. So catalyze, like for example, the reaction of the proteins. This is a good example. Like for example, this is like unphosphorylated protein. When this receptor is become active, so it can catalyze the reaction, and then this ATP will lose one of the phosphate, and then the losing one phosphate from ATP will become attached to this protein. So basically, this receptor after it's activated, so it can work as an enzyme to catalyze this reaction from ATP, and then protein become ADP, and then phosphate protein. So that's why it's called enzyme-linked receptor. That's because after it's bind to the ligand, conformation changed, and then this receptor will work as enzyme. And then most type of enzyme-linked receptor function as a protein kinase. What do you mean kinase? Kinase is enzyme that transfer the phosphate group from ATP to a specific amino acid in a protein. For example, tyrosine kinase attach phosphate to the amino acid tyrosine, where serine threonine kinase attach phosphate to the amino acid serine and threonine. So basically, this type of kinase enzyme, they always will attach the phosphate from ATP into a specific amino acid, make them then convert the amino acid into a phosphate form. So another one is called the G protein coupled receptor. This type of G protein coupled receptor, they found in the cell of the all eukaryotic species, are very common in animals. So this type of like G protein coupled receptors are very important for mammal. For example, the human genome has approximately 1,000 different genes that encoded like GPCR. So you can use some kind of term to describe G protein coupled receptor. So they play a lot of important roles on behavior, vision, and the sense of the smell. So let's talk about like G protein structure. GPCR, so like for example, this is GPCR protein. They typically contain like 7 transmembrane segment that win back and forth through the cell, through the plasma membrane. So basically, it's like this: 7, 1, 2, 3, 4, 5, 6, 7. This is 7. So they're like a fold together, and then the receptor interact with intracellular protein called the G protein. So like for example, this is G protein, that's the ligand. Why it called the G protein? Because this kind of a ligand, it can bind to the guanosine triphosphate and the guanosine diphosphate. So so why this G protein, like this ligand is called a G protein? Because this type of like protein is a ligand. It can bind easily to the GDP and the GTP. So GTP, the structure is very similar to the ATP, except like for example the base. One is G, another one is A. So and then this chapter shows how like receptor GPCR, how to react with G proteins. So like for example, this is G protein, and then so like first of all, the signal molecule bind to the GPCR, cause it to bind to the G protein, and then okay, so this is a G protein. This is like ligand, right? When the ligand bind to the GPCR, and then the conformation gonna change, right? Ligand, GPCR, and then when it's changed, it can extend, and then this extension will bind to those kind of G protein, right? And then the G protein will exchange the GDP for GDP. What's that mean? So so after this signaling bind to a receptor will bind to this G protein, and this G protein will like catalyze the GTP, right? And then GTP will lose one phosphate and generate the GTP, and then because the one phosphate okay, this is GDP so So GDP will insert inside of the GDP, and then this G protein will then leave this. They used to be bind, and then because of this this kind of like GTP insert inside of like G protein, so these two will disattach and dissociate, and then like they used to be like 1, 2, 3 units, right? And then this 1, 2, 3 unit were bound to this GPCR. After the GTP were bound one of the unit of their G protein, and then this whole thing, three units were dissociated with this GPCR, and then same time this one were separated with these two. And then this is called alpha subunit. This is beta and gamma dimer. So because this one was dissociated with the original 3 unit, so this is like activated subunit to promote the cellular response, right? And then the signaling molecule, and then will also dissociate from receptor, and then the up subunit, this small one, will hydrolyze the GDP into the GDP and phosphate, and then since like GDP already like converted into the GDP, so basically GTP will dissociate with this alpha unit, and then this like individual alpha unit will be released, and then it will go back to attach with beta and alpha, beta and gamma. So that's how it works. And then we're going to talk about ligand gated ion channels as another type of receptors. So how did it work? So ion channels, like for example, we've been talking about the ion channel when we're talking about the process, like how the cell membrane exchange the material between intracellular and intracellular space. So ion channels are proteins that allow diffuse of ion across the cell membrane. So basically, the iron, if they want to get inside of their cell, they will go through those kind of cell membrane. So this type of ligand-gated ion channels, also called ionotropic receptor. They belong to the third type of cell surface receptor found in the plasma membrane of animal, ant, and fungus. Like here is how it works. Then signaling molecule like ligand bind to this type of the receptor. And then ion channel, after it's binding, this ion channel will open and allow this iron go through the membrane and change the concentration of iron in the cell. So in animals, this type of like ion channel receptor are important in the transmission of signals between neurons and the muscle cells and between two neurons. For example, the acetylcholine receptor is ligand-gated ion channel receptor that's found in the skeletal muscle cell plus membrane. They can recognize this acetylcholine. Acetylcholine is a very important neurotransmitters released from the nerve cells. So when this neurotransmitter bind to these receptors, and then it will allow the sodium movement between the different cells. So if the cell will go through those kind of ions, it will trigger some kind of signals. So this is how cells they interact with each other. So by movement of this and ion generate some kind of signals. So cell also have some intracellular receptor activated by signaling molecule that's passed through the transmembrane. So before all the examples we've been talking about, it's just the receptors go through both extracellular space and intracellular space. Now we're going to talk about the receptors inside of the cell. So the three type of receptor I've been talking before belong to the they're located in transmembrane, but then some also can found inside of the cell. That's called the intracellular receptor. Under this kind of circumstance, our extracellular signaling molecule must diffuse through the plasma membrane to gain access to its receptor. So which means the receptor is inside. So signaling molecule have to be inside of the cell, then can binding with its receptor. So like for example, in some animals, steroid hormone receptor are intracellular. So like for example, some kind of intracellular receptor, they can regulate the gene transcription. So basically, those kind of receptor, they control how gene was transcribed. So like for example, take example as estrogen. This is some kind of steroid hormone. Because like for example, this is like estrogen receptor, right? And then it's inside of a nucleus. So estrogen first it's outside of the cell, and then first it has to go through the cell membrane, right? Go inside of the cell. Estrogen like diffuse across the plasma membrane, enter the nucleus, and when it enters the nucleus, it will bind to the estrogen receptor subunit. Then this subunit will go through some kind of like conformation change like this. Right. And then after it go through some kind of change, two receptors, they're gonna bind together like this, right? They form a dimer. And then this dimer will bind to like specific gene, right? And then this kind of binding to the specific gene, it will activate the transcription. And then since the transcription was activated, so from the DNA, we can get the mRNA. And the mRNA can be translated into the protein. Those kind of protein can have some kind of function to affect the structure and function of the cell. So basically, the intracellular receptor can regulate. Here's regulate gene protein. So here's a good example. Like this specific gene was transcribed after this kind of intracellular receptor binding with the ligand, and then start a transcription. Okay. So we're going to talk about receptor structure and the location and effect when they activated. So like, for example, we're talking about the four different type of receptor. Like first three, there's like the receptors, they go through membrane, and last type, it's inside of the cell, right? And then, for example, the enzyme-linked receptor, it's located in a plasma membrane. So how does it work? It's where phosphorylation of the intracellular protein, and then for the second one, GPCR, it's also located in the plasma membrane. It will bind to a G protein like here, right? After it's activated, it bind to this 3 subunit aggregation of G protein, and then after GDP bind to alpha subunit, and the alpha will dissociate with beta and gamma. And then after GTP were converted to the GDP, those three subunits can be reaggregated again. And then this one is ligand-gated ion channel. So basically it's also located in plasma membrane. It opens ion channel, right? And the last one would be intracellular receptor. It's located in the cytosol and nucleus. It can turn on the gene chain, specific genes transcription. Now we're going to talk about signal transduction and the cellular response. So like we said, we're going to talk for this chapter. We're gonna turn our attention to intracellular event, which we enable cell to respond to a signaling molecule that bind to a cell surface receptor. And then after its binding, it can introduce the signaling transduction and cellular response. So so like for example, we're gonna talk about tyrosine kinase. So this is one type of like a receptor. After ligand binding to this kinase, it will activate the signal transduction pathway, and then this kinase, protein kinase, can alter the gene transcription. So so this is how it works. So this is a ligand. It's called the EGF molecule, right? This is a ligand, and then we're binding to this kind of a receptor. This is two EGF receptor, like EGF called epidermal growth factor, right? And then after the two ligand bind to these two receptors, these two receptors will be dimer, so they will aggregate together. And at the same time, they will phosphate each other. So which means there will be additional several phosphate will added to their structure, and then GRB, this is another protein, will bind to like phosphorate receptor. GRB is protein will bind into this receptor, and then at the same time the GRB will bind into SOS. That's another protein, and then SOS will stimulate RS to release the GDP. And to bind the GDP. So basically this GDP will be released, and then after it's released, RS will change the conformation, and then this conformation will help it bind to the GDP will bind to the RS, and then after this binding, GDP bind to RS, this protein gonna activate another protein called RAF. And then this kind of binding will start a protein kinase cascade in which this new RF protein will phosphate another protein called MERK, and then MEK, MERK, and the MERK will then phosphate MERK phosphorate ERK, that's like a chain reaction. This one activated this one, and then this one will activate the MERK, and the MERK will get phosphate, and then this MERK will help to phosphate the ERK, ERK, and then this ERK become phosphate. So, and then this protein will enter the nucleus, and then it will phosphate the transcription factor this is transcription factor MYC and the FOS, and then both of them will be phosphate. And then this phosphate, MYC and the FOS, that's called transcription factors. They will stimulate the transcription of specific gene, maybe this gene, and then this RNA will be transcribed and then generate the new protein. So that's how this receptor, like ligand binding with the activation, and then finally go through a long pathway to promote or trigger some specific gene transcription and produce the new protein. So this is all I'm talking about. So we're going to talk about like one very important messenger that called cyclic AMP. So this is cyclic. AMP. So it's a very important component of many signaling transduction pathways. So this pathway also can be called signal transduction pathway, because ligand and the receptor binding and then trigger a long path. Finally, they transduced some kind of gene translation and transcription. So it's the same thing. So cyclic AMP also are very important for this kind of a transduction pathway. So let's talk about one good example about the signal transduction pathway and the cellular response involved in the GPCR. So like, for example, when the actual cellular signaling molecule bind to the cell surface receptor, that's called the first message, right? Because it's just the interaction between the actual cellular signaling molecule and the cell surface receptor. After this binding, first the message bind to the receptor, such as like most times it will bind to GPCR. And then this kind of binding will trigger the production of second message, small molecule or iron. That's relay signal inside of the cell. So basically the first binding, it will produce some kind of chemicals. And those kind of chemicals, that small molecule iron, then can trigger more signaling inside of the cell. So production is kind of a message called the second messenger. So this type of like generation of second messenger are very quick. It's very short. And then let's talk about how this second messenger, like cyclic AMP, it's a good example about one type of second messenger. We're going to talk about how it's produced. So like for example, if a ligand interact with GPCR is a big protein. It has several subunits. So one type of subunit is called adenyl cyclase, and this is enzyme in the plasma. And then if some kind of signal molecule are binding to this type of like enzymes, it's will trigger this type of enzyme to synthesize cyclic AMP. So this is a structure. So how did it happen? So basically it's generated from ATP. This is ATP, and then they use these enzymes. They generate, like, they get rid of 2-phosphate, and then they form some kind of like for example, here, this OH will attach to this phosphate and then get rid of these two. So basically building a new bond between O and the phosphate, and then cleave this bond and then and losing 2-phosphate. Then this is cyclic AMP. And then so this structure is CAMP. So we'll see how this CAMP will be used during like further on pathway. So we take the example. We take example with like when the hormone epinephrine interact with GPCR. GPCR, because this kind of epinephrine is some kind of molecule which generate under the stressful situation. It promotes cellular change, such as increase in ATP production and help the individual deal with the threat. Like for example, you got frightened by something, and then your body will generate epinephrine. And then those kind of epinephrine is like a signal molecule. It will help your body generate more ATP to respond to those kind of threat or danger. So we can call this kind of epinephrine. It's called a fight or flight hormone. So how does this thing work? So how does this the whole like pathway work? First, like epinephrine, we're binding to this GPCR, and this kind of binding will activate this kind of receptor, and then we activate GPCR, and then at the same time, we'll make this G protein. Then we'll promote this kind of G protein to bind to GTP. So basically, after it's activated, this G protein will bind into the GPCR, which this kind of binding will activate will help promote this G protein will binding to the GTP, and then after GTP binding to this kind of alpha subunit of the G protein, the beta and the gamma gonna dissociate from this whole G protein, and then after dissociation, this alpha subunit, it will also binding to this kind of enzyme called adenylyl cyclase, and this kind of binding will promote this reaction, ATP. This reaction will promote the ATP convert to the CAMP. Okay, and then generation of this is CAMP. We're binding to the pKa, right? This is another enzyme, and then this kind of binding will release the catalytic subunit of a pKa. So okay, so this is inactivated pKa. Used to be like have two subunit. One is like catalytic function, another one regulatory function, and then after the CAMP binding to this purple one, and then this yellow one will be released, and then so this yellow one called catalytic pK will be released, and then this released catalytic subunit, and then will phosphate some kind of specific like a protein inside of the cell, make it a phosphate, and then because due to this kind of phosphate protein, some kind of a reaction, cellular response will occur. So that's how like CAMP, how it was generated. It's generated from here, and like it generated from halfway here. So basically by like activation of this enzyme, and this is how CAMP was generated. And later step, it's CAMP was involved in the later stage to activate some specific like protein for cellular response. So okay, so let's see. We're gonna talk about a specific example, right? Like we said, this pKa, it's catalyzed, like stimulate the phosphorylation of the protein and then for further cell response. So we're gonna use here as an example to talk about like this step in further details. So look at this figure. It describes how skeleton muscle cells responds to the like higher level of epinephrine. So basically, like for example, you got frightened by something, your body generates hormone called epinephrine. So like, and then your whole body, how it responds to this kind of generation of the hormone. So this is a receptor, like for example, when pKa was activated, pKa activates this yellow stuff was activated, and it will phosphate two enzymes, right? Like this enzyme, phosphorylase kinase, it will activate this enzyme, and another one called glycogen synthesis enzyme. Both of enzymes get phosphate. So both of these phosphate enzyme, they were involved in the metabolism of glycogen. So why metabolism glycogen is important? Because glycogen is some kind of like energy, like glucose, like sugar glucose stored in the liver, right? When we are hungry, we need like our body to disassemble, like decompose glycogen and make glycogen to become glucose, sugar. Those kind of small sugar can be used by our body for energy. So that's how it works. Then when phosphorase kinase is phosphorylated, it's become activated. So these activated enzymes can be used to phosphorylate another enzyme in a cell called glycogen phosphorase. So this enzyme can be activate another enzyme and then make it be convert from inactivate into activate form. Then this enzyme become active, and then this enzyme can use by the body to cause the glycogen breakdown into like smaller glycogen unit. Like for example, used to have like n unit of glucose glycogen. Now with this activated enzyme, it's become like it's get rid of one of the glucose, right? Even though it's losing like what the one molecule of glucose which are losing will become phosphate, right? It's losing, and then this cycle can keep going with the use of glycogen phosphorylase. So another thing is like another enzyme for the glycogen synthase, right? This kind of enzyme was used to make glycogen, right? When this pKa was activated, it will convert activated like synthesis become to inactive way, like become phosphate. So the body will stop making a more glycogen, because right now, so basically this signal, this amplified signal just send it to our body saying we need more energy, right? How do we make the energy? We just hydrolyze the glycogen into the glucose, right? And then in the same way, it also will tell our body stop making the glycogen anymore. So it's this kind of amphiferin is basically inhibit the generation of the glycogen. At the same time, it will promote the decomposition of the glycogen. And so and then so like for example, if the ligand dissociate with the receptors, and the cellular response will be reversed. Like for example, so if the signaling molecule is no longer produced or its concentration become lower, a large percentage of the receptor are no longer bounded by their ligand. When a ligand dissociate from a GPCR, the GPCR become deactivated. So like for example, so that's how the body will happen. The like F subunit will hydrolyze its GTP into the GDP, and then the F subunit will reassociate with the other two dimers to form like a bigger G proteins. And then the amount of CAMP decreased due to the action of enzyme called phosphodiesterase, which convert CAMP into the CAMP. So the same thing, when CAMP level falls, the pKa will release CAMP, and then the yellow one and the yellow one. So it's like basically it's opposite from here. So like for example, if this ligand dissociate, it's coming off from the receptor will go back to its original conformation, and then like in this step, GTP alpha subunit, like it's dissociated with beta and gamma. Now it will aggregate together, become this like together, streamers together, and then at the same time, this yellow and the blue pKa, they will aggregate together and then become like original inactivated pKa. And then this CAMP will become less. CAMP will catalyzed by some kind of enzyme, convert to AMP. So CAMP will become less. So since we need CAMP for further down pathway, since the level of CAMP become less, so further down pathway will become less and less. So it's a reverse of cellular response. So why this second messenger is very important, because it can amplify the signal and the speed. What do I mean? Signal application. So basically, so during this kind of signal amplification, they involve the synthesis a lot of like CAMP, this molecule, right? And then with the more and more CAMP molecule coming out, and then more and more like activated pKa will generate, right? These kind of yellow things. So more yellow things, yellow subunit will dissociate with that purple one, right? Only this type of yellow one, individual separate dissociate yellow ones can be active the enzymes to phosphate, like more and more protein will be phosphate, right? If more and more this kind of protein will phosphate, it should generate more and more signals for cellular response. And then also, so this is good example about about the CAMP signal amplification examples. Then we're going to talk about like CMP's another advantage about the speed. Why the CMP can speed up the cellular response? That's because most of the times this CAMP molecule, they're very small, they are very soluble in the water, and then they can diffuse very easily through the cytosol. Like for example, and then like here give an example, say some scientists, they study like response, like neurons response to a signaling molecule. This is they call the serotonin, and this is a good neurotransmitter bound to the GPCR. And this small amount of this kind of small molecule ligand, they believe to play important role in depression, anxiety, and other behavior disorder. So how do they do? How do they like measure the CAMP level during this kind of experiment? So basically, the neuron cell, they grow neuron cell in a laboratory, and then this kind of neuron cell they inject with like fluorescence protein, and this kind of protein can change its fluorescence when CAMP is made. What that means is like you can measure the cell, the neural cells fluorescence fluorescence emission, and then based on the change of fluorescence emission inside of a neuron cell, you can estimate the amount of CAMP which were generated. So you can see from here, you can see, so like within 20 seconds, a huge amount of CAMP was generated after addition of the this kind of ligand. So basically, when you add those kind of ligand inside of a neuron cell, right? Because CAMP is the second messenger. As long as you add the ligand inside after 20 seconds, so before you add the ligand, there's no CAMP, right? Because we cannot see any fluorescent signal. It's just blue color. But after you add a signaling, a signal molecule, we see a huge number of like very strong fluorescence, which means a large number of like CAMP was generated. So like yellow color is intermediate level, and the purple color is a high level. So then we're talking about the crosstalk between different signaling transduction pathways. So which means, like, sometimes like several transduction pathway can cross each other. So what that means is when cells are exposed to multiple type of signaling molecule at the same time, the cellular response to these signals often depend on crosstalk, which is the phenomenon in which one or more components of one signal transduction pathway infect a different signal transduction pathway. So like, for example, some kind of ligand bind to a receptor, and then it triggers some kind of pathway. During this pathway, there are like more chemicals, more compound was generated, and then generation of the compound during the pathway, it will trigger another signaling transduction pathway occurring. So this is a good example explained how crosstalk among the three different signaling transduction pathway happened. Like for example, so we have like three receptor A, B, C. in like respectively different hormone X, Y, Z was combined with ABC individually. Like, for example, it can happen individually, right? Like, but they can cross-talk to each other. For example, during this pathway, second pathway, it generates some kind of like component, right? With this component, with generation of this kind of a protein, you can say component is small molecule protein. Anyway, like a component during the pathway. With the generation of this protein, it can activate the pathway 1 on the left, right? And then, like for example, there's like 1, 2, these 2 protein was generated during the third pathway, but maybe this pathway can inhibit this protein. This protein are necessary for this pathway go further, but with generation of this protein, maybe it will inhibit this protein, and then pathway 2 will stop going. So that's called a crosstalk. Okay, now we're going to talk about hormone signaling in multicellular organism. Let's see. Cellular. Okay, I think I can still talking about for this. And next one, okay. Next, I'm gonna skip multicellular this chapter. Our next class. So next class, so you won't be here, right? Your mom told me, right? You can be. So next class will be next Thursday, right? Okay. So next Thursday we can talk about some chemistry, about the biology stuff. Okay, let's keep going. Then we're gonna in this chapter we're gonna talk about how hormone in multicellular organism will generate like a lot different response. So you're gonna learn the type of cellular response caused by a hormone, and then each cell type they responds differently with particular hormone. And then first we're going to talk about that. Even for the same hormone, but cellular response, different cell will have different cellular response. For example, so like for example, we just give example, like you got scared, you generate some kind of hormone called ampifferine, right? Ampifferine have a different effect throughout our body. So one of the cellular response we learn is that it can trigger a breakdown of the glycogen in skeletal muscle cells, and then it will generate more ATP. And then, but what happened to the skeletal muscle cells when it meets the ampifferine. But like for example in lung, when ampifferine can trigger all the different response, right? And when the ampifferine will binding to some kind of receptor in the lung cells, the lung will relax the airway, allowing a person to take in more oxygen. Like in the heart, when ampifferine binding to some kind of heart cell receptors, it's gonna trigger the heart muscle cells, causing the heart to beat faster. So so this table said like amplifying the same hormone in different organs, it will have a different effect. Like for example, amplifying, if it's in the eye, it can dilate the pupils. In the gland, it can inhibit the production of saliva. And for the skeletal muscle, we said it's gonna help break down the glycogen, release glucose. For the skin, it can trick the blood vessel and stimulate the sweating. For the lung, it's going to relax airway. So more oxygen is taken. For the heart, it's going to increase the rate of a beating. So, we're going to talking about a very important concept, apoptosis. So how cell was dead, like cell dead, cell will kill themselves in a certain ways. So actually, apoptosis, it's one of the most dramatic response, like the cell communicate with each other. So like, for example, apoptosis, which is cell death, this kind of behavior actually is one type of cell communication. We can say it's programmed cell death. So how did it happen? So the cell first shrinks and become round due to the internal destruction of its nucleus and cytoskeleton. So first, the organelle inside got destroyed, and then the cell gets smaller, shrinked. And then plasma membrane then forms in regular extension, like this is plasma membrane. Before it's like round, now it's become like a thread. It's like extend. And then cell will become like a fragment like this. It's will break into several parts and then destroy the cell. This is called apoptosis. So here's some like important role about apoptosis. So people find out like during like embryonic development in animal, it's like embryonic, which means like how baby will grow inside of the cell, how baby will grow inside before it's born. Because the embryonic development in the cell, it need to sculpt the tissue in the organs. For example, so at the very beginning, the baby doesn't have the fingers. The fingers on like human being's hand are initially warped. It's like when baby was early inside, so they don't have like 5 fingers. They separate during the development because of the cells go through the apoptosis, right? Apoptosis is also very necessary in adult organism to maintain a proper number of cells in tissue and organs. So like, for example, for the adult, we don't grow anymore, right? So the amount of cell inside should be always keep constant, right? But like, for example, new cells will be generated. If no cell will die, we're gonna have more and more cells. That will cause a lot of problem for the organism. So in order to keep the constant number of cells inside, so we grow new cells at the same time, the cell die by apoptosis. So this apoptosis also get rid of cells, like old cell, which has already been warmed or infected by virus, right? Those kind of old cell or damaged cell, they are very dangerous. They are risky cells. If you don't get rid of them, they have very potential to become cancer cell. That's very bad for our body. So you need to rely on apoptosis to get rid of those kind of bad cell, old cells. So So, like for example, if if cells cannot go through like regular apoptosis, like I said, old cell, sick cell will keep going in an uncontrolled manner, right? This is will become cancer, right? Because and then if but apoptosis have to keep in a normal level, right? It cannot be like too slow. Too slow will cause cancer, but it cannot be quick, right? Too quick, cell die too quick will also causing like disease. Like one of example would be viral disease. Like for example, like HIV, right? AIDS, HIV, and then what's the how to say it? What's HIV? So basically, it's our immune cell. Like for example, there's a tons of like virus, bacteria around us, right? But like regular people, they don't get sick. Why? That's because we have a lot of immune cell inside of our body, for like T cell, B cell, some kind of white cell. There's several type of immune cell. Okay, if you want to learn immunology in the near future, you will get to know more than me. So because of this called immune cells, they help our body to build up good immunity, right? To fight against those kind of virus, bacteria outside, so we won't get sick. But HIV, it's like a virus. HIV will destroy those kind of immune cell inside of our body, because those kind of immune cells get destroyed because of HIV. So they cannot help us to defend outside of the virus. So people got sick, got infected by outside of the cell. That's called like viral. That's the reason of HIV. That's because with the HIV, this type of a virus, we make like an immune cell die too quick, right? And then this cause like disease. Another example would be neuron degenerative disease. It's like this type of disease because neuron cell like die too quick. So in our body, like in this kind of patients, they don't have enough neuron cell, right? Because all neuron cells are die, so they have this kind of disease. So for example, Parkinson disease, right? Because they are losing a lot of like neurons. So they have Parkinson disease. So they cannot feel regularly. They cannot control their action, their behavior regularly. That's called Parkinson disease. Okay, so we can talk about like a pathway due to the apoptosis. Like I just said, like apoptosis, it's also one type of like cell communications, right? And it's involved a long pathway to send the signal to the cell to say you should die. It's time for you to die. So this kind of apoptosis is one type of cellular communication. It involves the activation of cellular signals pathway. One pathway called the extrinsic pathway begin with the activation of death receptor on the cell surface. So basically, this is how it works. This is death receptor. This is death domain. It will bind to the extracellular molecule, and then this will initiate the pathway, right? So signal molecule is trimer, like three monomer that bind together, and then this trimer will bind to the death receptor. This kind of like trimer bind to receptor will causing those kind of death receptor, they aggregate together, and then so it will generate, because of the binding, it will generate some kind of death inducing signal complex. So this kind of complex. So it will generate those kind of blue or like yellow compound, right? Those blue and yellow compound were binding to this ligand and the receptors, form some kind of a signaling complex. And then, so this kind of initiator cascade will be cleaved from complex. So it will cleave here, right? And then this is like smaller active initiated cascade. This and this will keep, and a smaller molecule will be generated. Excuse me. And then because of the generation of this active initiated cascade, it will help to cleave this executioner pro-cascade, right? And then it will generate executioner cascade like this, right? This is active form. And then this executioner cascade will cleave the cellular protein. Like it's, for example, it will cut this kind of actin filament, and then the cell will shrink, and then eventually it will die. That's the process, how it happened. This is called extrinsic pathway. And then there's another pathway called apoptosis called intrinsic pathway or mitochondrial pathway. And it's triggered by DNA damage that would cause cancer. So this is called extrinsic because it's need extracellular signal molecule to trigger the generation of the pathway, right? This is called intrinsic. So which means the molecule inside of a cell will be the trigger to causing to trigger apoptosis. A good example is like inside DNA were damaged, and then the DNA damage will be the trigger, right, to cause the apoptosis. That's how it happened. Like for example, if DNA get damaged, mitochondria will release some kind of protein called cytochrome C into the cytosol, and then this protein will form a complex with other protein called form a complex called apoptosis. This apoptosis then will initiate the activation like this. Okay, so here's what we're talking about. For this chapter, we've been talking about cell communication, right? So we need some kind of signal, which can influence the property of the cell. And this signal molecule will bind to receptor to initiate a cellular response. And the cell signal will enable cells to sense and respond to the environmental change and communicate with other cells. So there are different types of cells and the cell communication, right? In terms of its mechanism and the distance between the cell and the cell communication. So cell communicate in like 5 ways, right? Direct intercellular, like cell and the cell, they direct interact with each other, they contact with each other, right? It's another way called autocrine, paracrine, and endocrine signaling. So cell signaling is usually like a 3-stage process involving like a receptor activation, signaling transduction, and cellular response. And the signal transduction pathway consists of a group of protein that convert initial signal into a different signal inside, right? We're talking about cellular receptors. there are like the type of the receptor, including the enzyme-linked receptor, GPCR receptor, ligand-gated ion channel, right? And then receptor, they are different structure and location and effect. And then we're talking about the signal transduction and the cellular response. How signaling pathway influence the cell divide. And we're talking about second messenger, CAMP, and how it's important play important role in the signal transduction pathway. And then we're talking about how CAMP was found in skeletal muscle cell. How does CAMP play role in the pathway in skeletal muscle cells? And we've been talking about like how hormone epiphone will initiate the cellular response in different organelle. And then we're talking about CMP can amplify and increase the speed of the signaling pathway. And then we're also talking about the crosstalk between the different cellular response. We're talking about hormone signaling in multicellular organelle, right? Like for example, epinephrine, they play different roles in different cells. Okay, we're also talking about apoptosis, how cell will die, what's the process, how does it happen, and what's the extrinsic and the intrinsic pathways, right? What's the difference? And then we're going to talk about. I will skip the chapter 10 since we only have how many lecture we have. I think we have 4 left, right? I can talk about more like genetic thing. This is very important, right? Because what is genetic? Genetic is a branch of biology deal with inheritance, right? So basically, how you get some characters from your parents. And first, we're gonna talk about some like chemistry about genetic material, especially DNA, right? If DNA is so important, it since DNA is very important genetic material, what's the structure? And also is a structure, we have to observe the DNA in the molecular and cellular level, right? We have to learn how DNA they replicate, how DNA was packed into a chromosome. How DNA was organized into the gene, right? How those genes are expressed at the molecule level to produce mRNA, protein, and non-coding RNA, right? And then we're going to talk about how genes expression was regulated. And we can talk about how mutation will change the property of genes and lead to the disease such as cancer, right? And then we're going to talking about how chromosomes are sorted and transmitted during the cell division, right? We're going to talk about relationship between the transmission of the genes and the outcome of the offspring's traits. We're going to talk about the genetic pattern as very important model called Mendelian inheritance model. And how does this model work? And we're going to talk about bacteria and virus, unique genetic information. And then we're going to talk about the role gene play in development of animal and plants from the fertilized egg to adult. And then we're going to talk about some genetic technology. And okay. Let's talk about it one by one. So what is DNA, right? DNA is genetic information, right? And then And then so DNA carries genetic informations for living organisms, right? So genetic material allow organisms to survive in their native environment. For example, an individual DNA provides the blueprint to produce enzymes that need to metabolize nutrient in food. So like for example, so which means DNA must have several like key criteria. It has to be contain information, right? The genetic material must contain the information necessary to construct an entire organism, right? A DNA genetic material can be accurate copied, right? Those process called DNA replication, and then it can also have the character of a transmission, right? After it's replicated, the genetic material must be like passed down from parents to offspring, right? It can also pass from cell to cell during the cell division, and then it's some kind of character of the variation, right? Difference in genetic material must account for known variation within each species and around different species. What else? Okay, then we're going to talk about the chromosome, right? So DNA was stored inside of a nucleus, but DNA is not like random. It's not separate. It's not individual. So basically, DNA have to complex with some kind of protein called keystone to form like DNA and the protein complex called a chromosome. Chromosome is a real like genetic material within the nucleus. We can talk about the chromosome, okay? And then most times when scientists they have to observe the genetic information, genetic DNA replications, they have to observe the change pattern about the chromosome during the cell division. Right. So okay, let's talk about some interesting stuff. Like for example, like in the early stage before like scientists they discover like gene, they open like before they open the area biological area called genetics. How did they like scientists find out those kind of genetic material? So this is what they did. So some kind of microbiologist, English microbiologist called Griffith. He's working on some type of a bacteria called pneumococcal. And so he found out like for this type of bacteria, there are several strains. So what do you mean strain? It's like for like pneumococcal, this is bacteria, but there are several different type, like type of other bacteria. Their property and their character very similar, but they are different, like behavior different. So which means their genetic information is different, right? One of their behavior they found out like one type of like pneumococcal bacteria, they can generate some kind of polysaccharide, right? Those kind of compound. But the other type of pneumococcal bacteria cannot. So which means why they are all belong to same type of bacteria, but they behave differently. So which means they have different genetic material inside. And then how did he do? So so like for example, in his experiment, he wants trying to like find find out what's different between S string and R string of pneumococcal. So what he did, so he inject like S pneumococcal into the mice, and then he found out all mice, all animal, all mouse and mice are died. Then he conclude this type of S type of pneumococcal, it must be virulent, so which means it's killing the animal. And then he inject another type called R pneumococcal into the mice. He found out like mice was alive. So he was thinking even R must be different from S. R is benign, so which means friendly, nice, not that toxic. Then he like and then he used the high temperature to kill the S, right, bacteria, and then he inject those kind of dead S bacteria into the mice. Mice still alive, so he's assumed like S died after with the high temperature, right? So it died. It cannot replicate and grow inside of a mice. That's why mice survive. And then he injected a combination of R and heat killed S into the mice, and then mice was died. So that's weird, right? You inject either R and heat killed S individually, and mice died. Why you put them together and mice life, but when you put them together, mice was dead. So he conclude maybe R has been transformed. It's converted into the toxic S, and from that heat killed S. So how to explain those kind of phenomena? So he called this transformation. So basically, S R, which like a benign R, can be transformed into the toxic S. So how does this like transformation happen between R and S? He assumed there must be some kind of unidentified material response for this kind of phenomena, right? And then based on those kind of phenomena, he conclude maybe there's something like genetic information, like from heat killed type S bacteria has been transformed to the life type R bacteria. So basically, even though S bacteria is died, right? But there's some kind of genetic material come out from the S, and then it's those kind of genetic material from that S transformed into the live R, and then those kind of genetic information will use R as a host. It will glow, and it will show the character of the toxic S. That's what is confirmed. That is assumed. So he assumed those kind of genetic information is the gene. It's the gene thing. Then we're going to talk about RNA and DNA structure. So I've been talking about like DNA and RNA structure a little bit before, right? We know like both DNA and RNA are all called nucleic acid, right? They are all like DNA and RNA. They are all polymer. They're like polymer, like protein. Protein is polymer. The only difference is protein. It's consists of like amino acid. Amino acid is protein's building block. But here for DNA and RNA, the building block is called the nucleotide, right? And then we also get to know DNA is a large, big molecule, right? It's composed of a small building block nucleotide. So So DNA is just formed by the convergent link of the nucleotide in linear manner. So basically, nucleotide is a monomer, right? DNA just along linear structures made up by the nucleotide. Made up by the nucleotide. And then DNA have double strand, right? Here's double strand. This is one strand. This is another strand, and they bond each other through the hydrogen bond. So in the DNA is a double helix structure, and then these two DNA strands, they are twisted around with each other from form a structure that's very similar to a spiral staircase. So DNA is associated with a different protein to form chromosome. So DNA is associated with the protein organized like this kind of long double helix into a compact structure. So a genome is a complete complement of organism's genetic material. For example, the genome of most bacteria is single cellular chromosome, where eukaryotic cells have DNA in their nucleus, mitochondria, and chloroplast. So let's talk about nucleotide structure. So this is so we can call like, for example, this is nucleotide, like this is structure, like this is like a building block for DNA called, it has three parts. Left part is called phosphate, right? This phosphate, and this is ribose. So in this position, there are only like one OH here. That's called the deoxyribose, and then this is a base, right? So this is like DNA's building blocks. So they only have ATCG, and this is like RNA nucleotide, right? They're the same, very similar. Only different is RNA's nucleotide is called ribose. What's different between ribose and deoxyribose? Ribose, they have two OH here, right? But deoxyribose only have one here, right? And then RNA nucleotide, they have AUCG, like this A, GC is the same. But remember, T, it's only existing in the DNA building blocks, and the U replace T existing in the RNA's nucleotide. So another thing is like DNA have like a double strand, right? These two strands, they coupled with each other through the hydrogen bond, right? So hydrogen bond, they match with each other. This C and the G, they will match with together to form the hydrogen bond between these two strands. And then A will form the hydrogen bond with T during this double strand. So you cannot mix with them. Always G with C, A and the T, okay? So let's talk about a little bit detail, right? So for example, when we're talking about deoxyribose, this is fine memory. And then label it from here. Okay, this carbon will bond to the base. Okay, you will always label this carbon which bond with base as number 1, and then it's number 2, number 3, number 4, number 5, and the carbon 5 will connect to phosphate, right? And then this is the base, right? We always, like, if we label this base rings, we always label the 1 connect to this carbon, the carbon on the ribose, deoxyribose as 1, and then go forward 1, 2, 3, 4, 5, 6. So remember, you have 3 parts: phosphate, base, and the ribose. So, okay, so we're going to talk about more for next class. Okay, I'll see you next Thursday. Okay, bye-bye.
Okay, see you. Thank you so much. Good night.